Hello everyone, welcome to the study of Chapter 2, Epigenetics. At this point, I will introduce Section 1, What is epigenetics? Finding the causes of many diseases only from the abnormal DNA sequences is one-sided, often with less effort, and you may never find the answer to some diseases. From central dogma, the information flow from genes is from the DNA genetic code to an intermediate RNA copy and then to the protein synthesized from the code. The process of copying genes or transcription of genetic code and producing proteins through translation of the code into chains of amino acids is called gene expression. In addition to genetic information, the genome also contains epigenetic information, that is, when, where, and how to apply genetic information. What is epigenetics? Let us take a study from this video.
Identical twins originate from the same DNA, so how can they turn out so different, even in traits that have a significant genetic component? For instance, why might one twin get heart disease at 55 while her sister runs marathons in perfect health? Nature versus nurture has a lot to do with it, but a deeper related answer can be found within something called epigenetics. That's the study of how DNA interacts with the multitude of smaller molecules found within cells, which can activate and deactivate genes. If you think of DNA as a recipe book, those molecules are largely what determine what gets cooked when. They aren't making any conscious choices themselves. Rather, their presence and concentration within cells makes the difference. So how does that work? Genes in DNA are expressed when they're read and transcribed into RNA, which is translated into proteins by structures called ribosomes. And proteins are much of what determines a cell's characteristics and function. Epigenetic changes can boost or interfere with the transcription of specific genes. The most common way interference happens is that DNA, or the proteins it's wrapped around, gets labeled with small chemical tags. The set of all of the chemical tags that are attached to the genome of a given cell is called the epigenome. Some of these, like a methyl group, inhibit gene expression by derailing the cellular transcription machinery or causing the DNA to coil more tightly, making it inaccessible. The gene is still there, but it's silent. Boosting transcription is essentially the opposite. Some chemical tags will unwind the DNA, making it easier to transcribe, which ramps up production of the associated protein. Epigenetic changes can survive cell division, which means that they could affect an organism for its entire life. Sometimes that's a good thing. Epigenetic changes are part of normal development. The cells in an embryo start with one master genome. As the cells divide, some genes are activated and others inhibited. Over time, through this epigenetic reprogramming, some cells develop into heart cells and others into liver cells. Each of the approximately 200 cell types in your body has essentially the same genome, but its own distinct epigenome. The epigenome also mediates a lifelong dialogue between genes and the environment. The chemical tags that turn genes on and off can be influenced by factors including diet, chemical exposure, and medication. The resulting epigenetic changes can eventually lead to disease if, for example, they turn off a gene that makes a tumor-suppressing protein. Environmentally induced epigenetic changes are part of the reason why genetically identical twins can grow up to have very different lives. As twins get older, their epigenomes diverge, affecting the way they age and their susceptibility to disease. Even social experiences can cause epigenetic changes. In one famous experiment, when mother rats weren't attentive enough to their pups, genes in the babies that help them manage stress were methylated and turned off. And it might not stop with that generation. Most epigenetic marks are erased when egg and sperm cells are formed. But now, researchers think that some of those imprints survive, passing those epigenetic traits on to the next generation. Your mother's or your father's experiences as a child or choices as adults could actually shape your own epigenome. But even though epigenetic changes are sticky, they're not necessarily permanent. A balanced lifestyle that includes a healthy diet, exercise, and avoiding exposure to contaminants may, in the long run, create a healthy epigenome.
After the show, we can summarize the following points. Epigenetics, a study of the chemical modification of specific genes or gene-associated proteins of an organism. Epigenetic modifications can define how the information in genes is expressed and used by cells. The epigenome comprises all of the chemical compounds that have been added to the entirety of one's DNA, that is genome, as a way to regulate the activity, also called expression, of all the genes within the genome. The chemical compounds of the epigenome are not part of the DNA sequence, but are all attached to DNA. Epigenetic modifications remain as cell divide and, in some cases, can be inherited through the generations. Environmental influences such as a person's diet and exposure to pollutants can also impact the epigenome. While genetic changes can alter which protein is made, epigenetic changes affect gene expression to turn genes on and off. Unlike genetic changes, epigenetic changes are reversible and don't change the DNA sequence, but they can change how your body reads a DNA sequence. So, epigenetics is a feedback loop between the genes and the environment. Our cells are constantly updating our genes on what they need to thrive. But inputting the correct environmental variables for the genes, food included, we can create health, longevity, and leave beneficial epigenetic marks for the next generation. Epigenetic coaching is all about optimizing the inputs to your system. Your DNA contains the recipe for making proteins. Your cells send signals to modify the epigenetic marks on your genes that tell RNA how to use the recipe and produce the correct proteins for the current environment. Some epigenetic marks are inherited and some are acquired. RNA reads DNA and helps assemble proteins. RNA and proteins can also be epigenetically modified directly. Then, proteins build cells, and cells build your body. Cells send enzymes to modify the ingredients they receive to match the environment. The environmental inputs create unique requirements for your cells. They act as sensors for what the current situation is. They might require different proteins to work with a specific environmental variable such as drugs, stress, food, exercise, toxin, and so on. Characteristics of epigenetic include variable, altered gene expression, and no DNA sequence changes. In epigenetic inheritance, mitotic inheritance is common. Intergenerational inheritance is occasional. Transgenerational inheritance is rare. So, we can't choose the genome, but we can make epigenome better by no smoking, healthy diet, exercise, and reading. For example, if you choose harmful diet, you will probably get the obese phenotype with the epigenetic change that don't alter your DNA sequence but affect the gene's expression. This can cause fatty liver diseases, altered aging, immune disorders, and cancer. In contrast, if you choose the safe diet, then you will get lean phenotype, delayed aging, and low disease risk from the corresponding epigenetic changes. The different diet choices will cause epigenetic alterations that not only have individual efforts but also can have transgenerational effects which are determined by epigenetic reprogramming and gametocarrier determinants. For a best phenotype, the offspring might get disease phenotype with increased risk of metabolic disorders, type 2 diabetes, cancer, or growth abnormalities. On the contrary, for lean phenotype, the offspring might get normal phenotype with reduced risk of disease. The types of epigenetic modifications refer to DNA methylation, histone modification, microRNA expression modulation, and etc. Epigenetic modifications change the genomic structure and thereby influence gene expression without modifying the underlying DNA sequence. Epigenetic modifications are controlled by a set of enzymes that can be functionally defined as writers that modify their target by adding residues, erasers that modify their target by removing residues, and readers that recognize and bind modified tickets and act as intermediates or partners in protein-protein interactions. DNA methylation can be transient and can change rapidly during lifespan of a cell organism, or it can be essentially permanent once set early in the development of embryo. Other likely permanent chemical modifications include histone acylation, ubiquitination, methylation, and phosphorylation. Macro-mediated genetic silencing plays an important role in gene expression. The specific location of a given chemical modification can also be important. For example, certain histone modifications distinguish actively expressed regions of the genome from regions that are not highly expressed. These modifications may correlate with chromosome binding patterns generated by staining procedures common in karyotype analysis. Similarly, specific histone modifications may distinguish actively expressed genes from genes that are poisoned for expression or genes that are expressed in different kinds of cells. That's all for this section. Thank you very much.